Hello guys, my name is Minhajul Islam and my topic is about the biological oxidation from the book M. Wolkenstein. Bioenergetics processes leading to the synthesis of ATP, the charging of biological accumulators take place in mitochondrial membranes. A spatially organized molecular system responsible for the energetics of living organisms are also localized in mitochondrial membranes. The synthesis of ATP in mitochondria is coupled to electrons and ion transport and to mechanochemical phenomena. The functions of mitochondrial membranes are very complicated and varied. Another type of bioenergetic is coupling membranes. The membrane of plant chloroplast is responsible for photosynthesis. The source of energy required by a living cell of all its various life functions is respiration, that is, the oxidation of organic compounds by the air oxygen. In 1780, Lavoisier demonstrated that respiration and combustion are of same nature. The investigations carried out of last 2 centuries by chemists, biologists, and physicists have led to the elucidation of the basic features of biological oxidation. The fuel, that is, the substances being oxidized, enters the animal's organisms together with food in the form of fats, carbohydrates, and proteins. These are broken down, that is, they are hydrolyzed in reaction catalyzed by special enzymes, that is, fatty acids. They are activated by means of enzymes and ATP are converted into acyl derivations of coenzyme A. The oxidation of acyl derivation of coenzyme A involves a number of steps, at each of which is there formed a residue of fatty acid containing two carbon atoms less than the previous one. The overall equation of the reaction of oxidation of a fatty acid with an even number of carbon atoms to acetyl CoA is as follows. Here NAD is the coenzyme, that is nicotinamide adenine dinucleotide. E stands for the enzyme. FAD is the coenzyme flavin adenine dinucleotide. The degradation of fats leads in the long run to the formation of acetyl CoA and also a propanil CoA and glycerol. The breakdown and oxidation of carbohydrates lead to the formation of triophosphate and pyruvic acid. When proteins are broken down, apart from individual amino acid, they are formed acetyl CoA, oxaloacetate, alpha-ketoglutarate, fumarate, and succinate. These processes of metabolism have been thoroughly studied in modern biochemistry. The compounds formed undergo further transformation in a cyclic sequence of a reaction known as citric acid cycle, the tricarboxylic acid cycle, or the Krebs cycle. The sequence of reaction is localized in mitochondria. The Krebs cycle is one revolution of the cycle consists of 8 reactions, involves the degradation of one molecule of acetyl CoA or one molecule of pyruvate with the formation of carbon dioxide and hydrogen, that is water, that is the burning of the molecule. The Krebs cycle starts the pathways of many biosynthetic reaction, the synthesis of a carbohydrate, lipids, purines, pyrimidines, and porphyrins. Protein synthesis is also associated with the cycle that produces the precursors of a number of amino acids. At the same time, a biological oxidation is the source of energy stored in the ATP. The reaction of the cycle gives rise to carbon dioxide and H positive ion. This is accompanied by the reduction of the coenzyme NAD and FAD. For a complete biological oxidation to be effected, these coenzymes must be oxidized. The electron transport chain is a polymerase system which accepts electrons from the Krebs cycle and the fatty acid oxidation cycle. Flavoproteins or enzymes containing flavin coenzyme FAD and flavin mononucleotide. Riboflavin 5-phosphate, the cytochromes contain a heme group, the iron atom of which undergoes oxidation and reduction during the operation of 2 cycles. The electron transport in the above chain occurs from left to right, terminating in the reduction of oxygen, which unites with hydrogen forming water. The electron released in oxidation unites with the next link of the cycle. The electron transport is coupled with the accumulation of energy in the molecules of ATP. In other words, the release free energy is converted into chemical energy of the ATP. Oxidative phosphorylation occurs. The most important phenomenon was discovered by Engelhard in 1930. Ballister and Sabakwa carried out the first determination of the PO ratio. that is the ratio of number of molecules on the esterified inorganic phosphate of that of the oxygen atom absorbed, and established the value of the coefficient is not less than 2. Phosphorylation occurs at three key steps of the cycle: NADH, flavoprotein, and cytochrome B. This provides the synthesis of at least 3 moles of ATP. The efficiency of the protein is equal to 92 by 211.3, that is about the 40%. The energy stored in the ATP is consumed by the cell of performance of chemical, electrical, transport, and mechanical work. A specific feature of oxidative phosphorylation that distinguishes it from enzymatic reaction in a solution in the special localization of the stages of a multi-step process. Oxidative phosphorylation is a localized in mitochondria and is directly associated with the functionality of their membranes. The oxidation reduction reaction briefly describes descriptive as a result of application of delicate chemical and physical methods. An important role was played, in particularly by the works of Chance, which was devoted by the spectroscopy of electron carriers, intact mitochondria. Probably these carriers of electron transport change are grouped into four complexes called the green complexes. The corresponding scheme is shown in figure point 13.4. The molecular mass of each of complexes is about 3 into 10 to the power 5. Such a complex is about 64% protein and 30% lipid. An electron transport complex is defined as the smallest unit in the electron transport chain that retains its ability to transport an electron. The structure and functional organization of mitochondria were the subjects of intensive investigation. Nevertheless, no solution has been so far obtained to many very important questions: the specific membrane structure of the mitochondria, the presence of an autonomic program of protein synthesis, that is, the DNA synthesis, and the mechanico chemical activity of the mitochondria that are directly associated with the role of power plants of the cell. Thank you.